Hi friends, my name is Akhilanand Singh. Welcome to the second video of the Archaeological Artifacts Dating series. In part 1 video, we talked about the radiometric methods. In this video, we'll be talking about the following 7 non-radiometric methods. Why do we need to learn about so many dating methods? We learn about multiple methods because no single method can work on samples of all the types. Environmental conditions at fossil sites are also different. They dictate what method works best under the given conditions. All dating techniques have limitations around datable range of time period. Some methods work best on artifacts belonging to the recent past, some are more suitable for samples billions of years old. Do I need to know all the technical details about different dating methods? No. Paleontologists, archaeologists and anthropologists need not know the detailed chemistry behind each method. They only need to have high-level understanding of things. Dating is done in highly specialized laboratories by trained physicists, chemists and other lab technicians. Let's begin with the trapped electron methods. Trapped electron dating is a collection of two absolute dating methods. These methods allow dating a crystalline material up to a few 100,000 years ago. All crystalline materials like quartz, feldspar and calcite have imperfections or defects in their structure. These defects have ability to trap electrons released from nearby radioactive elements and also the radiation from the cosmic rays. When artifacts like ancient pottery, bricks, rocks and human teeth are buried, they are exposed to radioactive elements in surrounding soil. The ionizing radiation from the cosmic rays also reach them. These materials keep accumulating electrons in their imperfections. The trapped electrons build up over time at a fixed rate. Measuring trapped electrons gives an idea about the time lapse since the material was buried. The time for which it was buried can be calculated by dividing the total absorbed radiation by the radiation rate. This fact is the basis of the trapped electron dating. There are two methods of measuring trapped electrons: the thermoluminescence method or the TL method and the optically stimulated luminescence method or the OSL method. Let's understand both these methods together. In the thermoluminescence method, a part of the sample is converted to grains and these grains are exposed to heat in a laboratory. Heat releases the trapped electrons in form of light. The quantity of released electrons is measured. Under the optically stimulated luminescence method, the crystalline material is exposed to the light of a specific wavelength. The light releases the trapped electrons which are measured. The light can be a visible light or an infrared light. The TL method has a disadvantage that it requires a significant portion of the artifact to be damaged. This can be a severe limitation in dating a precious ancient artwork. The OSL method doesn't cause such destruction. The OSL method is generally better, but its sample collection process is a little cumbersome. The sample must be kept in dark in a metal tube and must not be exposed to light while transporting from the collection site to the dating laboratory. The TL dating technique has been used to date bricks found from the Indus Valley civilization sites. It has also been used to date Neanderthal graves. The OSL method has been used to date artifacts in the Levant region, South and North Africa and Australia. The TL method was developed by Farrington, Daniels and colleagues in 1950s at the University of Wisconsin, USA. The OSL method development is credited to David Huntley and colleagues at the Simon Fraser University in Canada in the year 1984. On your screen you can see the image of the Mohenjo-daro site of Indus Valley civilization where the TL method was used. The images on the right hand side of the screen depict the sample collection process for the OSL method. Amino acid racimization method. When it comes to dating biological materials, there aren't many methods available. We learnt about carbon-14 dating method in the previous video, but it cannot date samples beyond 60,000 years old. The amino acid racimization method was introduced in 1980s and was refined throughout in the 1990s as an attempt to increase the variety of methods suitable for dating organic materials. Here are some key things about the amino acid racimization method. The amino acid racimization method is based on certain properties of amino acids. Amino acids are present in all plants and animals. There are 20 important amino acids. All of them except glycine can exist in two different forms called the L form and the D form. The full form of the L form is levorotatory and the full form of the D form is dextrorotatory. You can see the L and D forms image on the screen. These forms can be differentiated using polarized light. All organisms when they are alive have only L-type amino acids in their bodies. When they die, The L-type amino acids begin transforming into D-type at a steady rate. This transformation goes on until there are equal number of L and D types. The ratio of these two types in a dead organic material is used to determine the time passed since the organism died. You might be wondering what is the meaning of the term racimization in this method's name. When a compound has only L or D forms of the amino acid, it is called an optically active compound. When it has equal number of both the forms, it is called an optically inactive compound. In chemistry, racimization is the process of conversion of the optically active compound into optically inactive one. Datable range of the amino acid racimization method. The datable range for this method depends on temperature at the site. It also depends on the type of amino acid, humidity, acidity and aridity. The process of racimization occurs much faster in the warm and wet conditions compared to the cold and dry conditions. The datable range at the sites having mean annual temperature of 30 degrees Celsius have the maximum range of 200,000 years. The datable range at the sites having the mean annual temperature of 10 degrees Celsius have the maximum datable range of up to 2 million years. The sites where the mean annual temperature is minus 10 degrees Celsius, the maximum datable range is up to 10 million years. Obsidian hydration method. Obsidian hydration method is used to date artifacts made of obsidian. Obsidian is a volcanic rock. It has shiny glass-like surface. The obsidian rock absorbs water at a well-defined rate when it is exposed to environment. The longer the artifact has been exposed, the thicker the hydration band will be. Early humans used to make tools of obsidian. These tools include flints, stone knives, projectile points, etc. All such tools are good candidates for dating using obsidian hydration method. To measure the hydration band, a small slice of material is cut from the artifact. Then this cut part is ground to make it 30 micrometers thick. This piece is then placed under a power microscope to measure the water content in it. The datable range for the obsidian hydration method is up to 200,000 years. If the artifacts made of obsidian have been discovered from a place where the mean temperature is high, the dating results might be erroneous. The obsidian hydration dating method has been extensively used in the prehistoric sites in North America, South America, Pacific Islands, New Zealand and the Middle East. The paleomagnetism dating method is based on the direction of Earth's magnetic field in the past. Earth's magnetic poles are different from its geographical poles. The magnetic poles keep shifting their positions and direction. Sometimes they even get reversed. Earth's magnetic pole reversal occurrences are quite random. In the last 83 million years, there have been at least 183 pole reversals. The latest pole reversal occurred 780,000 years ago. Certain magnetic minerals in rocks have ability to record the direction of Earth's magnetic field at the time they are formed. The same phenomena is witnessed in the magnetic material inside clay pots. When a clay pot is heated, the magnetic material inside it aligns itself along the magnetic field of Earth present at the point of time and location. On cooling, the magnetic elements get frozen. Since the position of Earth's magnetic direction in past are known to the scientists, the age of rock can be determined by studying the magnetic field and direction inside the magnetic materials. Fission track dating. This method is based on the principle of counting the number of tracks formed when uranium decays through fission and damages a crystalline structure. The damaged trails or tracks formed by fission are typically 1 to 20 micrometers long. The rate of decay of uranium isotopes are well known. This rate stays constant through time. The fission tracks in a rock are counted by an optical microscope, and then the age of the rock is determined based on the half-life of the uranium isotope. This dating technique is applied to date the rock in which fossils are found, not on the fossil itself. The datable range for the fission track dating technique is quite long. A sample having an age from a few thousand years to a few billion years can be dated using this method. This technique has been successfully used to date hard minerals like zirconium as old as 2 billion years. This method has been used to date rocks in South and East Africa. Electron spin resonance method. This method was developed in 1970s to date recently found materials that cannot be dated using radiocarbon methods. It has been used to date buried human teeth, animal cells and burnt flint in ancient ceramics. The method is very popular for dating animal teeth because teeth are the most common part of animal fossils. The minerals in tooth enamel possess electrons in two states, the non-excited state and the excited state. Natural geological radiation transforms electrons between these two states at a fixed rate. The ratio of electrons trapped in both the states is proportional to the time the sample was buried. This fact allows to find out the age of a sample such as a human tooth by counting the electrons changing states and applying the rate of change of the state. Time to conclude this video. We discussed about 7 methods of absolute dating which are non-radiometric. On the screen you can see the time range for various dating methods. The next video of this series will focus on relative dating methods. Till then, bye.